Please welcome Dr. Omar Cornejo from UC Santa Cruz to the stage.
Thank you, David. Actually, message from the sponsors. I think I'm going to give the talk in Spanish now, so vamos a empezar a hablar en español. That's not true. And the other one is that if I ever mention AI, I should acknowledge Terry in the backstage. Another thing is that if you were here for Chris Ballmer's talk yesterday, if you're from UCSC, you're supposed to mention the genome browser. I am not mentioning the genome browser, but I just mentioned it, so I've covered my bases. Thank you, PacBio, and thank you all for listening to this. I'm going to present some of our new work in the development of a capture method for sequencing the HLA. First of all, I want to acknowledge the people that have contributed to the development of the method. MCE, Mushin Yi, our lab manager, she is the master in the wet lab, and she has optimized all of our protocols in the wet lab. Matt Glassenab and Obed Garcia, postdoctoral scholars who have done the heavy lifting of the bioinformatic analysis with my unavoidable intrusions. I cannot help it, right? So why the HLA? The HLA, the Human Leukocyte Antigen region of the genome, is the major histocompatibility complex of the human genome. This is the region of the genome, 3.6 megabases, that encode for a very large number of genes that are involved in our immune system, especially genes that are responsible for our ability to differentiate self from non-self. And a lot of the effort in the sequencing and characterization of the genetic variation in the locus have been focused on HLA class 1 and class 2. There are three classes, class 1, class 2, class 3, especially on the classic HLA genes, which are the genes that are responsible for presenting peptides to T cells and the rest of the immune cells that will respond and mount immune responses. So why does it matter? It matters because alleles in the HLA are associated with increased susceptibility or increased resistance to infectious diseases, viruses, bacterial infections, parasites, but also different alleles in these genes are associated with the development of autoimmune diseases, right? In fact, the HLA region of the genome is a region that shows the largest number of significant association with traits in the genome. This is not a GWAS plot. It's just a plot that shows the number of significant associations in the y-axis along the genome. What is interesting about this, though, is that the majority of the associations are not necessarily with classical HLA genes or genes that are commonly found in autoimmune or related to autoimmune diseases. Something else that I wanted to mention, in this slide is that the class 3 region has long been neglected and is a region that doesn't have that much characterization, which is something that we also want to address in our methods. Another thing that is really important about the HLA is that HLA mismatches are associated with increased organ transplant rejection. As the number of mismatches increases in alleles of the HLA, the relative risk of rejection of transplants increases. HLA is highly polymorphic. It's the most polymorphic region of the genome. If we walk along chromosomes 5 and chromosome 6 depicted there, and we look at the amount of genetic variation that is present in different parts of the chromosomes, you will find in your walk a blip with a huge increase in genetic diversity, and that region corresponds to the MHC. So not only it is a region with the highest amount of polymorphism in the human genome, but also the polymorphism is not evenly distributed across different parts of the genes that make up the HLA. So the majority of these SNVs are present in exons of the coding part of the sequence of some of the proteins, the classical proteins of the HLA, that encode for the domains of the protein that present peptides, right? That's not a surprise because our immune system and our HLA genes have evolved to be able to adapt and present different peptides during the course of evolution, right? So this leads to a problem. This high diversity and these high levels of polymorphism have left to us with a conundrum, which is the high uncertainty in the identification of genetic changes in that region, right? And actually, this is evidence in those plots here. One of the things that I want to bring your attention to is that when we are talking about characterizing the HLA, we not only want to identify SNVs, and if a SNV is heterozygous or not, or different SNVs are heterozygous or not, we want to know how those SNVs are arrayed linearly to make a protein. Because ultimately, that combination of those mutations is what I'm going to derive in functional differences of what those alleles are doing. So funnily enough, if we actually start looking at our ability to impute alleles in different populations from different parts of the world, our ability to impute alleles accurately ranges from 20% to, well, sometimes 100%. But, and not funnily, right, the populations that are less accurate, for which we have less accuracy, are populations that are traditionally less studied, right? South American natives, North American natives, Oceanians, right? So this has to change. And one of the things that I want to emphasize again here is that we need to identify the combination, the linear combination of mutations that make up a protein. In the field of immunogenetics, which is actually new to me, I'm a population geneticist by training, the standard is the identification of alleles as this combination of mutations that are inherited together from the father or your mother, right? And they make up this funny nomenclature of alleles. The funny nomenclature is actually pretty useful because we can then correlate the alleles that we have with functional information that has been clinically derived to then think about alleles that are protective, alleles that are increase the susceptibility to infections, and so on and so forth. And these alleles have a very specific way of describing. So they have 4 fields, and the first field corresponds to the serotype, the immunological information contained in the allele. The second field corresponds to the non-synonymous changes that are present in that allele. The third field, the synonymous changes, and the fourth field, the non-coding changes that are present there. And all of them seem to be relevant to really describe these alleles and find then their functional implication in disease. There are many challenges associated with genotyping of the HLA, and this is its evolution, baby. This is the result of evolutionary processes. HLA genes evolve via duplication and diversification of the genes, and it's believed that selective pressures have contributed to, yes, the generation of polymorphism in the region, but also conservation. So the function of presenting peptide is maintained in many of these different genes. The end result of this is that the distance, sometimes the similarity between different genes, make it really hard to use short-read data, for example, to identify accurately SNVs in these genes, right? What you can see here is the overlap between HLA-C domains, for example, with exons in HLA-B. They're very difficult to recognize and differentiate one from the other. Other challenges in the identification of alleles in the HLA have to do with reference bias. This is work that Diogo Meyer has done, a Brazilian colleague that has worked with HLA data for a long time from the 1000 genome, and he has shown how there is a strong, in standard pipelines of analysis with short-read data, there is a strong bias to maintain reference alleles. And this is the consequence also of the way we treat short-read data. Remember, I told you that the majority of these changes are located in specific exons. So we're going to have short reads at times that have a very large edit distance between the reference and the read. And usually what happens to those reads is that they get poor mapping quality. And they will contribute very little then to the identification of new alleles. In addition to that, we have the issue that we want to know not only what changes are present in those alleles, but how they are arranged linearly, right? So we want the phasing information. And structural variation, large insert sizes between, large intron sizes between certain exons makes this process of phasing of those alleles very difficult. given there are more problems associated to this, like the structural variance in the region and so on and so forth. Given all of these problems and our interest in solving some issues associated with the association between the vaginal microbiome and the genetics of women, we were interested in actually developing new tools to characterize the HLA. That took us to develop a new method to capture and sequence with long-read sequencing technologies the HLA region of the genome. So we, in collaboration with Twist, developed a new set of probes to expand the set of probes that they have and include class 3 genes. And then standardized protocols to multiplex, pool, and prepare libraries for sequencing with long-read technology. And we use both PacBio and ONT, and I'll show you results from both. We also work on standardizing our bioinformatic analysis to select the best tools that produced the most high-quality data that we could get. And that actually took a while, but finally we got to a nice set of tools. And we developed a new program to actually identify HLA alleles from our highly resolved haplotypes of the HLA. To benchmark the tools and to benchmark the protocols, we took 2 data sets. One data set consisted of 16 geographically diverse samples from the Human Pangenome Reference Consortium. The reason to get these samples to do this is because we have the true haplotypes. We have access to T2T diploid assemblies for all of these samples, so we could compare the full haplotype length from our HLA to what is supposed to be the truth. Also, we included 4 genomes that are part of that set of the genome in a bottle to assess our accuracy in genotyping. In addition to that, I told you one of the end goals of this process is to identify alleles as per the IGDP nomenclature. So we included 16 geographically diverse samples from the International Histocompatibility Working Group to assess our ability to identify alleles. First, very general high-level results. We get very high, much higher average read length with PacBio than ONT in our capture, about 4 kilobases long. And we get, with both technologies, high-fold enrichment of the HLA. The on-target versus off-target ratio is very high, with very high coverage, hundreds of X coverage for each one of the samples. And another thing that was encouraging is that there don't seem to be any biases from the technologies. They seem to be mapping, and the coverage seems to be pretty consistent between the two. I told you we developed probes to try to target a larger number of genes than what is available currently from either commercial protocols or the most up-to-date academic protocols out there. In fact, we are bringing the HLA genes that we're capturing up to 82 genes at the moment. And we can produce very high coverage and specificity in the sequencing of both class 1, class 2, class 3 genes. What you see here is a coverage plot only with primary alignments of reads that go on each one of those islands of genes, with the oceans of very low coverage, almost no coverage, in the intergenic regions. And the class 3 region is a very densely packed region of the genome, and we are actually able to sequence the majority of the region, and we get gaps where we expect to see those gaps because of the design of our probes as well. So the high specificity in the primary alignment to these regions is very important to us. Why? I told you something that was really, really important about the HLA, and this is not only true for class 1 genes, which is high homology between many of those genes make it very difficult to identify accurately mutations, right? So we can show that, an example case, right, that we can accurately map with primary alignment the gene CYP21A2 that encodes a 21 hydroxylase that is responsible for a synthesis of cortisol, and the deficiency, mutations that cause a deficiency in this gene are involved in congenital adrenal hyperplasia. So if you have short-read data, and you try to map that short-read data with some of those mutations to this region, what is going to end up happening is, because of the high homology of that gene with a pseudogene that is located downstream from the gene, leads often enough to misidentification of variants. And with our method, we can actually differentiate very clearly those. In fact, this region is even more interesting than that. We have one sample from a Puerto Rican individual that has a C4b deletion. He's heterozygous for the C4b deletion, which is that orange gene that you find in between those two genes. And C4b deletions are commonly associated with lupus. So this region and the resolution of this kind of structural variants, as well as the accurate mapping to different locations, is an essential thing for us. So I told you it's essential, so we need to measure how good are we doing, right? How well are we doing in actually identifying variants in the HLA region. So when we look at genome in a bottle, we were really pleased to see that we get very high accuracy in our identification of SNVs. The gene concordance, the genotype concordance, between our newly sequenced data and what is considered to be the true set is really high for PacBio for the identification of SNVs, and at clinical level up to higher than 99%, and we have very high accuracy on the identification of indels. This is not the case for ONT data. Now, this is really important because for our downstream analysis and the identification of alleles, this has an impact, right? We cannot trust the calls that we're getting from ONT as much as we are trusting our PacBio data. The next step is, okay, we can do well in genotype concordance. We get really high quality genotypes, but can we resolve the haplotypes? That's the next level. So there are two levels of problems in this question. The first level is, can we put together haplotypes? And how much of the expected haplotype can we recover? So this heat map here shows you that we are doing really well. So for the most part, we are able to recover full haplotypes for all of the genes in class 1, the classical genes in class 1 and class 2 regions, with the exception of very well-known cases. DRB1 is a complicated gene, as we all know. People that work in the spectrum know. And the next step of all of this is, okay, we can extend our phase and recapitulate our haplotypes, but are those haplotypes accurate? Meaning, can I bring the accuracy that I'm getting at the genotype level down to the haplotype level, right? And show that we're getting very accurate haplotypes? Yes, we do. Right, this is, again, a heat map that shows the edit distance between our newly sequenced haplotypes and the expected from the HPR, the Human Pangenome Reference Consortium haplotypes. The boxes in gray correspond to haplotypes that are either missing in the reference or are homozygous for the alleles. So this is great, because we are showing that we're capturing effectively, efficiently the HLA genes, enriching in an effective way, in an efficient way, the sequencing for the HLA, and we're getting very high accuracy at the genetic and haplotype level at recovering HLA alleles. So the next step was, at this point, can we then identify HLA alleles as per the IMGT nomenclature? So usually what is done in these cases is that we get sequence data and we compare a newly sequenced data with a database that has the sequence data that correspond to each one of the alleles that have been described to date. As you heard yesterday from Elisa, this database is not really that great. It's going to have to be revised at some point, but it's the only tool that we have to compare and to validate some of the things that we have. So there are many tools, and the state-of-the-art tools for the identification of alleles in HLA have been developed to deal with short-read data, not really with long-read data, right? When we use some of these tools and we pass our highly accurate haplotypes through some of these tools, we start seeing a bunch of different things that are super weird. For example, not surprisingly, we see a very large fraction of ONT alleles that are discordant between ONT and the true. PacBio effectively recovers the HLA allele that is expected from the IMGT, Then we also see cases where PacBio and ONT are discordant to the true, but they are concordant with each other. So we seem to be capturing the same allele with both ONT and PacBio, but they don't meet what the true should be. And we have cases where all three are discordant. So when we start looking in detail into some of these, and we use, again, state-of-the-art tools to characterize the alleles and to identify the alleles like spec HLA, we get cases like this. HLA-C, one of the haplotypes, is PacBio is identifying as C15, ONT is identifying as C12, but actually the allele, as per the first field of resolution, is as C17. This is a problem, right? So many of these tools, again, were developed for short-read data, not really for long-read data, and we had to adapt and create a new tool to make better use of our better resolved haplotypes to assign alleles. And we came up with HLA-Resolve. HLA-Resolve accurately assigns these alleles to the correct allele that we anticipate from the database, and we find in some cases, some mismatches that we consider probably wrong in the database, and we are much better able to resolve with PacBio data. The next steps for all of this is going to be how can we bring down and increase affordability of our method. Right now, we can prepare and sequence samples for about $100 per sample with this method, but remember, we have very high coverage, 100x coverage, right? So we did a downsampling experiment, and we can show that if we downsample the sequencing effort to 8 to 10% of the current sequencing effort, we can achieve comparable accuracy in the genotype call, both at the SNVs and the indels. And we think that with additional modification of the chemistry that we're working on right now and sequencing effort, we can bring the assays to have about $35 per sample to characterize the entire HLA, not only classic HLA genes, right? With that, I just want to thank all of the people that have contributed to this. A special thanks to Noel Bittner, who has supported us from the beginning of all of this and believed in us when we came and said, you know, we're not an HLA lab, but we're interested in doing this, and we need to solve this problem. And she said, yeah, it sounds reasonable. Let's do it. With that, I'm open to any questions that you have.